In this video, we're going to talk about the carrying of carbon dioxide by blood. Certainly, we've already discussed the carrying of oxygen and how that is really dependent on the expression of hemoglobin, right? Remember that almost 99% of oxygen carrying in the blood is bound to hemoglobin. And the adaptive significance of that is that oxygen has relatively low solubility in plasma or even the cytoplasm of the red blood cells. And so you can't dissolve an enormous amount of oxygen just in the liquids. It has to be bound to a carrier to really increase the carrying capacity of blood. And that was really the emphasis on hemoglobin and the dynamics of hemoglobin and the amount of hemoglobin to really enhance that carrying of oxygen. Now, we might focus on oxygen as sort of like the most important thing that blood is doing. It often is portrayed that way. However, we really can't forget about the importance of carrying carbon dioxide, because carbon dioxide, as we'll see in a moment, can lead to dramatic pH shifts very quickly. And pH is one of those things that we're really sensitive to, changes in pH, largely because of that marginal stability theory of protein structure that we talked about previously, wherein free protons, or a change in the concentration of free protons, which of course is indicated by a change in pH, can lead to dramatic changes in protein structure and function, which of course we want to avoid, right? The proteins are the gears in our metabolic machine, and so having them become dysfunctional would be very bad very quickly. So let's talk a little bit about that CO2 carrying. All right, well, CO2 is carried in three forms, as opposed to really two forms for oxygen. And really, you could almost narrow it down to one, right? Remember, it's sort of 99% bound to hemoglobin and 1% dissolved for oxygen. The three forms that carbon dioxide is carried in, actually two of them match up with the oxygen carrying mechanisms, right? So one of them is dissolved, and as in other words, as CO2. It's not modified. It's just in the solution. Now, carbon dioxide, as we mentioned before when we talked about Henry's law, carbon dioxide has much greater solubility in liquids than does oxygen. And so we can actually carry a fair amount of carbon dioxide just dissolved. And so instead of it being, sort of 1.5% as oxygen is of the total that's carried, we can actually carry about, let's just put a number of about 10% on it, sort of 5 to 10%, but we'll round up, right? And again, these are approximate values, and actually they differ a little bit based upon where you are in the bloodstream, because different areas of the bloodstream have different amounts of carbon dioxide in it. And again, as we'll see in a moment. All right, so the dissolved CO2 is carrying a reasonably significant fraction. Another form is bound to hemoglobin, right? As we just said, sort of matches up with the oxygen carrying. And that's actually called carbaminohemoglobin. And it's binding to particular side groups of the amino acids in the hemoglobin chain. We actually talked about this as well before, because when we talked about oxygen carrying, because when carbon dioxide binds to hemoglobin, it actually changes the affinity for oxygen. Remember, that was quite adaptive, because remember, our tissues down here the cells of our body are undergoing metabolism, and they're breaking down nutrients in the process of doing that, and they're releasing the carbon within those nutrients as carbon dioxide, right? And again, so that's some of that carbon dioxide, and it's a very lipophilic molecule, so doesn't have to go between the cells. Some of it will end up dissolved into plasma, right? Small fraction there. Some of it will end up bound to the hemoglobin here as carbaminohemoglobin, HbCO2. And remember, because it's binding that carbon dioxide, the hemoglobin affinity for oxygen is actually going to fall a little bit, actually aids the unloading, right? So the more CO2 we produce, indicating a higher metabolic rate and a greater need for oxygen, the more the hemoglobin will release the oxygen in response to binding this carbon dioxide. So we could actually add in, sort of as a byproduct of this, an increase in O2 delivery, right? Remember, that was part of the Bohr effect, right? Bohr, write his name up here, right? Bohr effect is this enhanced unloading of oxygen in response to carbon dioxide binding and actually free proton binding, right? And again, we discussed those previously. Okay, so those are two forms. Oh, and sorry, the carbaminohemoglobin accounts for probably, and again, I'm sort of generalizing these numbers, we'll call it about 20% of carbon dioxide carrying. So really, we're not even up to a third yet, right? So our other 70% is in the form of bicarbonate. Right? That's bicarbonate. And it is an ion. Some people say bicarbonate ion, but a lot of people just say bicarbonate. And the bicarbonate is formed. So if you think about this, there's a C and an O, and there's at least two of the O's, right? So there's carbon dioxide sort of within the structure of this ion. So how do we get to this ion? Well, the carbon dioxide that's diffusing in here, I have another arrow coming into the red blood cell, right, is going to be hydrated, meaning it's going to react with water with the aid of an enzyme. And it would react with it anyway, but the enzyme facilitates that reaction. And so we end up with the bicarbonate, the carbonic acid equation. Right? So here's our carbon dioxide that enters our red blood cell. There's water in there. It's going to react with that water to form an acid called carbonic acid. And there's the enzyme would be here. It's called carbonic anhydrase. Right? So that sort of aids this reaction, this conversion of hydration of carbon dioxide into this acid. Well, the acid dissociates pretty readily, meaning it comes apart, right, to form individual ions, which are H plus bicarbonate. Right? So our carbon dioxide is now within this bicarbonate ion. And again, what's going to drive the reaction, right? See how these double-headed arrows here mean that the reaction can go in either direction. But it's going to be driven inside the red blood cells here at the tissue. It's going to be driven in this direction. Right? So at the tissues, the reaction is driven in this way because the CO2 is going to be high. It's going to be elevated. And something called the law of mass action, or sometimes known as Le Chatelier's principle, essentially says that if there are more reactants on one side of a reversible reaction, it tends to go toward the other side, right? We add more of these things, There's plenty of water, right? So this is already in excess. But if we add more carbon dioxide, it becomes more likely to combine with the water and drive the reaction to the right, which is exactly what happens at the tissues, because remember, that's where the carbon dioxide is going to be highest, because that's where it's being produced. Okay? Now, remember that this H plus has the potential to be buffered by the hemoglobin molecule, right? And many, many copies of hemoglobin, right? And remember, that is another portion of the Bohr effect, right? Remember, we had the first, I'm going to draw an arrow here, from the HbCO2, right? That was also Bohr, right? So Bohr kind of has two components to it, meaning that when you bind, it binds, the hemoglobin binds carbon dioxide, or it binds a free proton, it will reduce the affinity and aid the unloading of oxygen that we indicated right here, right? And again, that's about O2 delivery. It's separate from this, but they're linked, right? The Bohr effect is linked to this enhancement of carrying as bicarbonate, which again is really important, right? We have to get all this carbon dioxide out of the tissues. Otherwise, we're going to end up with a pH problem at those tissues, right? Because if we can't get the carbon dioxide out, we still drive this reaction to the right. And notice here are these free protons, which would reduce the pH. Aha! But many of them, as they're being produced here, are going to contribute to the Bohr effect and be buffered by hemoglobin, limiting the pH change. Okay, so that's Bohr. But the Bohr effect and the Haldane effect are sort of linked. So the way you want to think about this is, because this H plus, well, let's go back to this law of mass action for a second, right? So at the tissues, the reaction is being driven to the right. Now these two are starting to accumulate. What's to keep these two from recombining and driving it back to the left? Nothing, right? But check it out. We're taking the H plus away, right? We're physically removing it because it's becoming bound to the hemoglobin, and of course the blood's moving, so it's carrying it away. But we're taking it out of solution, right, by binding it, which means it can't recombine with the bicarbonate and drive you backwards. That's called the Haldane effect, right? So this enhanced carrying of bicarbonate due to the buffering of the free protons, right, getting these away so we don't go backwards, enhances the stability of this bicarbonate ion and increases the carrying of carbon dioxide as this ion, right? We don't want to rely on, if it starts to go backwards, then we have to rely on it carrying as CO2. Well, can we carry lots of CO2? It's got good solubility, but it doesn't have fantastic solubility, right? And so our CO2 carrying as a whole would go down if we didn't have the Haldane effect, right? Once again, the enhanced carrying of bicarbonate due to the buffering of these protons by hemoglobin. Okay, now the last thing I want to point out is that this all is going to get reversed at the lungs, right? So let's redraw our reaction here. Right? And now let's envision ourselves at the lungs. Same acid in the middle here. Getting out of room here. Right? Now we're at the lungs. Okay, so just think of sort of exhaling at the lungs, right, regarding CO2. So what's going to happen to the CO2? At the lungs, the CO2 is going to go down, right, because we're getting rid of it, right? There's still some, this dissolved CO2 can immediately come out of solution. Okay, well, that's going to favor it coming off of the hemoglobin. That's going to favor this reaction now moving in this direction, right? If we're reducing the reactants on this side, then we're going to favor the entire reaction moving in this direction. Right? So this would be at the lungs. Right? And so now the bicarbonate content is going down, the CO2 content is rising, but remember we're exhaling it, right? So we're reconverting our bicarbonate into CO2 So it can be exhaled. You can't exhale bicarbonate. We enhance the carrying as bicarbonate, right, through the mechanisms we just described above, but we can't exhale the carbon dioxide within this bicarbonate ion if we reconvert it into the CO2, right? And so the H plus is going to come off the hemoglobin. That's going to mean that hemoglobin has a higher affinity for oxygen now, right? Leftward shift of that O2 dissociation, right? Go back to the Bohr effect, a leftward Bohr shift at the lungs, right? Of that curve. And again, that means that's good, right? Because you want high affinity for oxygen at the lungs. It's where we want to pick it up, right? So that as this reaction is drawn to the left, the H plus, right, will decrease. It will come off the hemoglobin. The bicarbonate will go down and be reconverted to CO2, and we will exhale this CO2 that we originally produced over here at the tissues, right? And so we've come full circle now. We know the three levels or three forms of carrying of carbon dioxide. We can see how they get into the blood and how they're carried here. But remember that at the lungs, we have to sort of reverse the direction, right? That CO2 has to come out of the blood and go into the alveolar air, right? The exact opposite direction of all the arrows I have here at the tissues. All right, and that's carbon dioxide carrying by the blood.